The overall goal of applying these chromatin immunoprecipitation approaches to differentiating embryonic stem cells or cardiac embryonic tissues is to uncover the epigenetic regulation of cardiac development.
Since fine-tuning of gene transcription during embryonic cardiac development is regulated by epigenetic components, chromatin immunoprecipitation is a suitable approach to understanding this process. The main advantage of this technique is that we can specifically target a locus and look at the epigenetic marks that sit on that specific piece of DNA.
For this method can provide insight into the epigenetic regulation of cardiac development. It can also be applied to other organs or any cell types. This is Imen. She's a PhD student in the lab, and she will do the experiment today.
This procedure utilizes embryonic stem or ES cells, embryoid bodies generated from ES cells, and embryonic heart tissues dissected from E9.5 mouse embryos. Fix the cells and tissues. Use 1% formaldehyde in PBS for the cells and PB2 permeabilization buffer for the embryonic tissues. Place the tubes on an orbital shaker at a speed of 60 rpm at room temperature for exactly 10 minutes. Stop the cross-linking reaction by adding glycine to a final concentration of 125 millimolar and incubate for 5 minutes at room temperature on the orbital shaker at a speed of 60 rpm.